So, have you heard of recombinant DNA technology before? If not, then let me tell you something about it. Recombinant DNA technology is the process joining together of DNA molecules from two different sources and inserting them into a host organism to generate product for human use. What is the use of this, you might ask? Well, it can be used for gene mapping, which is to define a map of the human genome, and it can help introducing useful information in the definition of human diseases. Next, the production of antibiotics. Genetically modified fungi have been developed to greatly increase the yield of antibiotics. Next is the production of hormone insulin. Genetically engineered bacteria used for production of insulin in exchange of the animal-made insulin can make sure people don't get allergy reaction. And the tools for recombinant DNA technology are as follows. First, you need a target gene or a gene of interest. Next is the restriction enzyme. You also need a host cell and a modifying enzyme, and the last one, you need a DNA cloning vector.
I will continue this topic with the tools used in recombinant DNA technology. So firstly, a target DNA, or known as a DNA that carries the gene of interest, where a desired part of the DNA sequence on original DNA to be cloned, that has to be cut out and transferred or inserted into a vector. Next, we proceed with the restriction enzyme, where the enzyme cuts the DNA molecules at the restriction site and produce short restriction fragments. But how does the restriction site enzymes work? So, it works by making two incisions on sugar phosphate backbone that has 4 to 8 nucleotides that are palindromic. To form these cuts, a restriction enzymes are used. For sticky ends, we use EcoR1 that are produced by E. coli. Will recognize the base sequence 5' to 3'. It makes staggered cuts within the sequence, which then produce sticky ends. And for the blunt end, a SMA1 enzyme are used. This enzyme will recognize the base above. The cuts will mate between the adjacent C and G.
DNA cloning factor. DNA molecule which can carry fragment into a cell and replicate there. There are three characteristics of DNA cloning factor, which is it is able to accept foreign DNA in MCS and able to replicate freely in the host cell origin of replication, and possesses selectable genetic marker that is resistant to antibiotics, for example MPR.
Examples of DNA cloning vectors are plasmid, bacteriophage, cosmid, and yeast artificial chromosome. Plasmid, DNA molecule that can carry foreign DNA into a host cell and replicate there. Capacity to carry insertion size is 1 to 10 kilobase. Bacteriophage, a virus that infects bacteria. Capacity to carry insertion size is 8 to 20 kilobase. Cosmid, hybrid vector, combination of plasmid and cosmid or fish DNA from bacteriophage. Cloning capacity is 35 to 50 kilobase. Yeast artificial chromosome, fragment of DNA from the human genome inserted into a yeast cell. Cloning capacity is 100 to 1000 kilobase. Host cell is a cell which is able to accept foreign DNA and allows the vector to multiply of recombinant DNA. Characteristics are able to receive recombinant DNA through transformation process, able to maintain the structure of recombinant DNA from one generation to another, able to amplify the gene product from the recombinant DNA.
And bacteria is the most commonly used as host cell. The reason is that the DNA can be easily isolated from or introduced into a bacterium. There are two types of modifying enzyme. The first one is DNA ligase. It seals the foreign DNA fragments containing gene of interest and bacterial plasmid. It also catalyzes the formation of phosphodiester bond to form recombinant DNA. Next is the Taq polymerase, which is used in polymerase chain reaction, and it replaces the DNA polymerase due to the temperature conditions of PCR. The function is to add nucleotides to the 3' end of primer in order to form new DNA strand. Next is the steps in gene cloning. Firstly, isolation of gene. Isolate the plasmid from bacterial cell and isolate the gene of interest from the DNA of the donor cell. Then, cleave or cut process will take place. So, the donor DNA and plasmid bacterial is cleaved using compatible restriction site. Site of cleaving is a single cut within the LAC-Z gene is made. And next is the insertion. The DNA fragment or gene of interest is inserted into the plasmid vector. It is sealed together by DNA ligase, thus forms recombinant DNA. Next is the transformation. So the recombinant DNA then inserted into the bacteria through transformation process. After that, amplification process will take place. As the bacteria reproduce, the recombinant DNA is also replicated or cloned. After it is being replicated, it will go through screening, which is to identify bacteria clone containing recombinant DNA, and blue-white screening is done.
In blue-white screening, we plate the bacteria in agar containing antibiotic ampicillin and excess sugar. After place it in agar that contain it, we can see that some recombinant DNA are resistant towards antibiotic ampicillin and others are not resistant and stop growing. The recombinant DNA that are resistant to antibiotic, they were divided into two colonies, blue colonies and white colonies. In blue colonies, the plasmid do not have recombinant DNA. Recombinant plasmid contain intact LAC-Z genes are able to produce the enzyme beta-galactosidase, which can hydrolyze X-gal. And in white colonies, the plasmid do not have recombinant DNA and the LAC-Z is not functional.